Hello, everyone. Thank you for joining us for tonight's webinar, Gene Therapy Breakthroughs for Cardiomyopathies, Insights into Tenaya's Clinical Trials. We're so glad you could be with us. We have tremendous interest in tonight's webinar. We have 235 people registered at last count, so people are very excited to hear Tenaya's amazing news. So let's get started. Next slide, please. First, a few housekeeping items. All attendee microphones are on mute upon joining so that we can hear the presenters. Please enter your questions into the Q&A feature of Zoom at any time, and we will have a dedicated Q&A session at the end of the presentation. If there is a question that's very specific to a certain slide, I may pop on and ask that at that moment, but most of them we will hold until the end. Next slide. Just a quick note, the DCM Foundation and the Genetic Cardiomyopathy Awareness Consortium do not endorse or encourage you to participate in any specific research study or therapy clinical trial. Given this disease has no known cure, we aim to provide this information to patients to review and decide independently if they would like to participate. If you are considering participating in a research study or clinical trial, please consult your medical team. Thank you. Next slide. So poll number one, let's push forward poll number one. We'd love to know who is attending tonight's webinar. Are you best described as a cardiomyopathy patient, family member or friend, a medical professional, or an industry professional? It'll be interesting to see the results. I know there's tremendous interest in this topic tonight. Excellent. If we can share the poll results. So we have about 37% are patients, and then another 3% friends and family, medical professionals at 17%, and industry professionals at 43%, which is not too surprising that everyone is interested in this news. So let's keep rolling. Next slide. So, and I forgot to introduce myself. I'm Jayanne Rock, the Director of Information and Education for the DCM Foundation. And I am thrilled tonight that we could have Tenaya Therapeutics here with us. Next slide. I'm excited to introduce you to Dr. Whit Tingley. Whit is an MD, Ph.D. He's the Chief Medical Officer with Tenaya Therapeutics. Whit joined Tenaya as the Chief Medical Officer in December 2018. And previously, he served at Cytokinetics as Vice President of Clinical Development, Cardiology, and at Genentech as Therapeutic Area Lead for Cardiovascular and Metabolism Research and Early Development. Between 2009 and 2017, he held Medical Director and Group Medical Director roles with increasing responsibility at Genentech and Roche. Whit formerly was Adjunct Associate Professor of Medicine in the Cardiology Division at the University of California, San Francisco, and cared for patients as an attending cardiologist there in their cardiology faculty practice. Whit earned a BA degree from Brown University and his MD and PhD degrees from Johns Hopkins University School of Medicine. Without further ado, we are just thrilled you could be here tonight, Whit, and so excited about this updated information you're going to share with us this evening. So take it away, and Whit's going to share in his own slide deck.
Yeah, thank you, Jayanne. It's an honor to be back presenting again at the DCM Foundation, and it's been terrific to see how you and Greg and others have grown the foundation and the impact that you're having for patients. Very much appreciated. So today we'll talk about insights from our clinical trials for gene therapies for genetic forms of cardiomyopathy. We'll have a brief introduction about Tenaya as a company. We'll talk about AAV-based gene therapy, how it works. We'll focus a bit on the MyPeak-1 study, a first-in-human study of gene therapies for hypertrophic cardiomyopathy due to mutations in the MYBPC3 gene. We had a release of early interim data from that study and I can share so I can share more details with everyone here today than we've been able to present before yesterday. And then we'll talk about the RIDGE-1 study, first in human study of a gene therapy for ARVC due to mutations in the PKP2 gene, and talk more about how you can connect with us and get more information. So Tenaya is 100% focused on heart disease, and our mission is to transform and extend lives through the discovery, development, and delivery of potentially curative therapies that target the underlying cause of disease. Today, we're talking about genetic heart disease and potentially curative gene therapies. We are fully committed to creating new therapies that address the underlying cause of genetic disease. And we currently have 5 clinical trials, including 2 active gene therapy studies, which we'll focus on today, MyPeak-1 and RIDGE-1, but also natural history studies that are important for understanding how diseases can affect people differently over time. So MyClimb focuses on children with HCM due to MYBPC3, and RIDGE focuses on ARVC, arrhythmogenic right ventricular cardiomyopathy, specifically for people with PKP2 gene mutations. Years ago, I presented at the DCM Foundation, and we discussed all the ways that patients can participate in the development of new therapeutic options. And I'm proud to say that we've been building that internally here at Tenaya. We're very proud that Wendy Borsari is on our team. She is Associate Director of Patient Advocacy at Tenaya, and also herself lives with genetic hypertrophic cardiomyopathy. We rely on other patients to let us know if we're doing what needs to be done and headed in the right direction to do what patients need next. We have a patient advisory council who we lean on frequently for advice about the design of our clinical studies, what the endpoints are, how to communicate what the meanings of endpoints are, our informed consent forms, is that appropriate and understandable? Is the study burden for patients appropriate? And all those topics. So we welcome your input and we'll continue to seek it out. If you have interest in contributing, please contact us at patientadvocacy@tenayathera.com. If you yourself want more information about HCM studies or ARVC studies, the links are shown on this slide. Of course, there are many other opportunities for patients to participate in drug development, including participating in interactions with the FDA. They are very interested in the patient perspective, and they incorporate that into their thinking about when the time is right to approve a new medication. All right, gene therapy. The gene therapies we're talking about are still investigational treatments for genetic heart conditions. We designed these to specifically express a working gene in the heart muscle to restore heart function and halt disease. We're targeting the underlying cause of the condition, a non-functional gene. So in both cases, we're talking about a variant in the DNA spelling that prevents the gene from working properly, and the goal is to replace that with a new fully functional copy. The ultimate hopeful outcome is that we improve heart rhythm, cardiac output, reduce symptoms, improve exercise capacity, and of course, quality of life. Both TN201 and TN401 are single-time IV therapy. So a single infusion, the gene is delivered, and then the expectation is that its effect will last for many years. A question we get a lot, and importantly, these gene therapies do not change the genes in your egg or sperm cells. So you do not pass the treatment on to your children. All right, here's a little more detail on how this works in practice. We take advantage of a virus, a type of virus called AAV, for adeno-associated virus. These are spread naturally among people, but don't cause disease by themselves. So they're a great tool to use. but we don't really use the virus. All we're using is the protein part of the virus, the packaging, if you will. We take out all the DNA components of the virus so that it's no longer able to replicate or behave like a virus. We're just using that protein as a package or a box to deliver the new gene, the working gene we're trying to replace, to the heart. Again, it's a one-time IV infusion. It goes through out the body. It will get taken up by many different tissues, but it's designed to turn on just in the heart. So this will bind the outside of the heart cell, which will then take it up inside the cell and bring it to the nucleus where the DNA is. The protein is broken down. The DNA then is added to the nucleus where it can start to make functional protein. We'll go back to how this all works in subsequent slides. There have been many successes in AAV-based gene therapies. The FDA and the EMA have approved 6 gene therapies for diseases such as spinal muscular atrophy, genetic eye diseases, 2 forms of hemophilia, Duchenne's muscular dystrophy, so forth and so on. And there's extensive clinical experience. Over 5,000 patients have been treated in AAV-based gene therapy trials, and over 3,700 patients have been treated with a particular gene therapy called Zolgensma for spinal muscular atrophy worldwide. Our gene therapies, TN201 and TN401, use the same form or subtype of AAV as Zolgensma, specifically AAV number 9. Of course, there are risks and potential risks with gene therapy. The immune system recognizes this as foreign and can react to it. So we use a preventative regimen of immunosuppression, including steroids such as prednisone, to prevent the immune system from overreacting to the gene therapy. We're trying to prevent it from rejecting the gene therapy so that the gene therapy can work. As part of this, the inflammatory or immune response to the delivery, there can be inflammation in the liver, which we detect with blood tests. These respond very much to the steroids like prednisone, so we titrate the dose of the prednisone to control any liver inflammation. Currently, it's a one-time dose, which is a big advantage. You get it and you don't have to think about it again. But there isn't an opportunity, is not an opportunity for a second dose in the future. It cannot be redosed. This is because even with the steroids and immunosuppression, the immune system will eventually develop antibodies that prevent that AAV delivery protein from working again. The AAV gene therapy can deliver genes to different cell types in the body, different tissues. We know that it does, including the liver. We use a specific component in the DNA called a promoter so that the gene only turns on in the heart cells. Of course, AAV gene therapies, including TN201 and TN401, are still under investigation. Not all the risks are known. All right, we're focused now on hypertrophic cardiomyopathy caused by mutations in the MYBPC3 gene and the TN201 gene therapy that we are developing. We've shared this data before, but just to summarize, this therapy works very well in animal models of the disease. So mice with mutations in the same gene develop hypertrophied hearts with dysfunction, and they die prematurely. But treatment with this gene therapy in the mice reduces the heart mass, reduces the hypertrophy, improves the heart function, and increases survival. So we are happy to be enrolling in the first-in-human study, a phase 1b/2 study of TN201. The study is called MyPeak-1. The study currently focuses on adults. In the future, we will develop this for children affected as well. But the current study focuses on adults. To be eligible, you have to have a mutation in the MYBPC3 gene, pathogenic or likely pathogenic mutation. but you can have either obstructive or non-obstructive forms of hypertrophic cardiomyopathy. You need to have mild to moderate symptoms of heart failure and low levels of neutralizing antibodies. We've talked about how the immune system can form these antibodies. Some people have them already, and unfortunately, if you have high levels of them, you're not eligible for the clinical trial. The primary goal of the phase 1b/2 study is to assess the safety of the gene therapy. We are comparing different doses in terms of both safety and efficacy, and we're exploring its effects on quality of life and signs and symptoms of the disease. We have many enrolling centers throughout the United States. You can see a list here. You can also find more information on hcmstudies.com or at the clinicaltrials.gov website, and the trial number is listed below. We're making good progress in this study. We have enrolled the first dose cohort of patients, and we can talk about initial data from these patients today. In October, importantly, an independent panel of experts called the DSMB reviewed all the data, safety and initial efficacy data, and approved an increase in the dose based on the appropriate safety profile. We have increased to the next dose level, and we are currently screening patients for that second cohort. We also implemented a protocol amendment that will increase access to the trial from more patients, for more patients. So an ICD, an implantable defibrillator, is no longer required. We increased the upper age limit to 75 years of age, and we opened it up to obstructive as well as non-obstructive disease. So the data that we released yesterday as preliminary data from the first 3 participants, and we are very pleased to say TN-201 has been well tolerated to date, that the DNA we're delivering has reached the heart as intended and is expressing RNA, and we'll talk about what that means. Levels of the TN-201 RNA and the protein product, which is called MYBPC, have increased over time. In terms of clinical endpoints, still early. We do see encouraging signs of improvement in some measures. Other measures are still too early or still mixed. So the clinical effects of TN-201 will become clearer with data from more patients over time, and including both dose levels. This is the baseline information from the first 3 patients enrolled who were very severely affected, more severely than the typical HCM patient. So in this table, the gray column shows a picture of what sort of an average HCM patient looks like in terms of age and procedures and biomarkers and so forth. And then we list these 3 patients and their status on the right. So the 3 patients are younger than the average patient, and they've had significant interventions for their disease. All 3 have had an implantable defibrillator at young ages, and all 3 have had open heart surgery to remove some of the hypertrophied heart. So an aggressive, important procedure. They had this at young age again, and so they have an aggressive form of the disease. Their heart stress biomarkers are elevated. The amount of hypertrophy is very high for hypertrophic cardiomyopathy, and they have daily limitations in terms of symptoms. So the initial safety data that we're getting is as we would expect for an AAV gene therapy, no unexpected findings at this point. For safety findings that are associated directly with the gene therapy, what we're seeing is the low-grade liver inflammation that we talked about before. These respond very well to steroid treatment. So we monitor frequently in all patients and then adjust the steroids if needed, if there's any amount of liver inflammation, just to get it fully under control again. Again, this is very typical of gene therapies. As I said, for example, Zolgensma has been used in over 3,000 patients, and these liver function changes are frequently associated with that therapy, but there have been no long-term effects such as liver damage. Importantly, we are not seeing concerning immune reactions such as one specifically named thrombotic microangiopathy. This typically or can occur at higher doses than we are using. So we are very happy that we aren't seeing any evidence of that so far in this trial. There's been no sign of heart toxicity, no heart inflammation, which is called myocarditis, no abnormal heart rhythms, and as I mentioned, we have removed the requirement for the implantable cardiodefibrillator. And the heart contraction strength has been stable, the ejection fraction, for those that know that terminology. All participants continue in the study, and the protocol includes 5 years of follow-up. There, of course, have been other health events unrelated to the gene therapy. The majority of these were mild and short in duration. There were 2 serious health events that occurred due to causes that unrelated to TN-201 itself. All right, so I promise we get back to kind of the mechanism and how this works in more detail, the DNA, the RNA, and the protein parts of gene therapy. We're using an analogy here in which, maybe you're hosting a party, you notice you don't have enough chairs for your dining room table. And so you decide to make more chairs. So you order a book, a manual in order to build the chairs. So the chairs here are the protein. The heart doesn't have enough MyBPC3 proteins and needs to make more. So the protein is the chair. So what you do is you order a manual on how to make furniture, and that comes shipped in a box. And the box is the AAV protein shell that we use for the delivery. The manual is the DNA, so the instructions. The box is delivered to your house. It's the IV infusion. The house is your heart cell. And then you bring the manual into your house to access the instructions. And that's the transcytosis of the vector into the heart muscle cells and the transduction by which the DNA is delivered to the nucleus. Now, the manual has instructions for many pieces of furniture. You specifically need that type of chair. So what you do is you make a copy of that particular instruction for that chair, so you can bring just the copy to your workshop. So that's the DNA being converted into mRNA transcription or mRNA expression. And so for the gene therapy to work, we need to see the messenger RNA being made. And we'll show you data soon about that actually happening in our study. So those instructions then you bring to your workshop, and you get all the pieces to build the chair. Those would be amino acids, and then you build the chair using the instructions. And so you build several chairs, you've restored the normal protein level in your heart, and you can have normal heart function. So in these trials, we measure all 3 components. We see if the DNA or the manual has been delivered to the heart, to see if it's expressed in terms of making the mRNA, and then see if that is then translated into protein. And we watch to see whether the protein levels increase. So we're very encouraged by the initial findings. I'll share findings from the first 2 patients, but the 3rd patient is not far enough along to have these results yet. So is the DNA getting to the heart? Yes, we're very encouraged that we're getting substantial amounts of DNA into the heart from these patients. We are doing endomyocardial biopsies, a procedure that's commonly used after a heart transplant, for example, to take just the smallest piece of the myocardium from these patients and analyze it. We can detect that the gene therapy is being expressed. So we do see RNA specifically made off of the DNA that we've delivered with TN-201. And we, in the first patient, we have sequential biopsies, more than one, and we can see that the protein level is increasing. So very encouraging initial data. Here, I promised we'd talk specifically about the RNA expression level. And we are encouraged by the levels that we're getting that is very similar to other AAV-based gene therapies and is increasing over time. So in this graph, you can see the level of RNA being expressed in the heart of patient 1, 8 weeks after the treatment, and 1 year after the treatment, or 52 weeks, and in the second patient, 8 weeks after the treatment. So clearly, the DNA is getting to the right cells in the heart, and cells are expressing the messenger RNA. And it is increasing over time. So patient 1, the level goes up by 50% between week 8 and week 52. These levels, as you can see, compared to the plum box, are very comparable to other gene therapies for the heart. One example of which you may have heard about is gene therapy for the disease called Danon disease. So we are achieving very similar levels to that gene therapy as well. And I'm happy to say that gene therapy has an encouraging initial data from their first in-human study, and they have fully enrolled their pivotal study, the study that could support approval of that gene therapy by the FDA. We look forward to those results, and we are encouraged to be in the same ballpark. At the same time, I do need to note that we are not at the level of RNA expression that we saw in mice with the disease when given the same dose. That's shown here in the green bar. So we anticipate that with more time and with the higher dose level, we will be able to bring our RNA expression higher. That may translate into additional efficacy. We don't yet know how much RNA is required to restore normal heart function. But with more patients, both doses, that's exactly what we expect to learn from this study. So again, just to be completely clear, we do not know whether this dose is the most effective dose or an effective dose. And while we compare to other gene therapies, including the Danon one I mentioned, that we cannot conclude that we will have efficacy or improvements in the disease based on that comparison. The diseases are different, the genes are different.
Well, we do have one question that's appropriate for this slide. If you don't mind, why does the expression increase over time? And how does the RNA or protein level compare to healthy patients without HCM?
Yes, so the process of delivering the DNA and then expressing it does take time. So in terms of delivery, that virus has to get to the cell has to take it in, bring it to the nucleus, unwrap it, form double-stranded DNA, and then express that DNA into RNA. And that process does take time. So the 8-week time point, we're not yet at steady state. So that process is still increasing at 8 weeks and increases through 52 weeks here. We don't know yet whether 52 weeks is the plateau, the maximum, or whether that will continue to increase even more than that. In this case, what we're measuring is only RNA made by TN-201. So we know it was 0 before treatment. We didn't do a baseline biopsy. We don't have the data for that, but because we've tested the assay in tissue from other heart samples, we know it's 0 at baseline. So all of this is new RNA from TN-201. And again, we don't know how much will be enough, but we look forward to gathering more data.
Excellent. Okay, thank you, Whit.
So this is still very early in the trial, and we're very pleased where we are. We are collecting clinical data, and the short answer is it's too early to say whether there are clinical benefits at this point. There are some encouraging signs. So we do look at biomarkers from blood work to signs of strain on the heart called NT-proBNP and troponin I. We see some signs of stabilization, and there is some evidence of improvement for patient 2 at week 40. But again, it's early and not definitive. We use echocardiography to look at the heart and the amount of hypertrophy and the improvements in the filling of the heart called diastolic function. Again, some signs of improvements, but not all consistently green here. So more time is needed. We're very happy that both patients are feeling better in terms of the NYHA, which is the New York Heart Association class, which measures how the disease affects daily activities. And so we're glad to see improvements there. The KCCQ, Kansas City Cardiomyopathy Questionnaire, is for patient reported symptoms, and we certainly need more time to see improvements there. So overall, we are encouraged by where we are. Over time, we expect to see more consistent improvement across multiple parameters. Just one slide to say that we are also still enrolling the natural history study that I mentioned before, MyClimb, focusing on children with HCM due to MYBPC3 mutations. We've had a lot of enthusiasm for this study. We've enrolled over 200 children. We're getting close to our goal of 250. And so encourage people to participate in clinical trials if you're able to do so. All right, I'll pivot now to the TN-401 gene therapy for ARVC due to mutations in the PKP2 gene. The design of the gene therapy is very similar. It also uses AAV9, the delivery mechanism, and so forth is all very much the same. So I won't repeat any of that. As with TN-201, we have extremely encouraging data from a mouse model of ARVC. So here I'm showing EKGs from mice. You can see a nice uniform typical heart rhythm from a healthy mouse. Mice lacking PKP2 function have abnormal heart rhythms. They have premature ventricular contractions and non-sustained ventricular tachycardia that are occurring throughout, so very easy to measure. When we treat with the mouse version of the gene therapy, we restore normal heart rhythm. We also show that we're restoring normal protein levels of the PKP2 protein. We're preventing fibrosis or scarring of the heart and slowing disease progression, and importantly, restoring a normal lifespan of the mice. So our phase 1b/2 study for PKP2 is called RIDGE-1, again focusing on adults, this time with PKP2 mutations. To qualify for the trial, you need to have frequent premature ventricular contractions on your EKG, and you must have a functioning implantable cardioverter defibrillator. And again, no low neutralizing antibodies to the gene therapy. As with the other trial, the main goal is to assess the safety or comparing 2 different doses and looking at the early effects on signs and symptoms of the disease. This slide lists the sites that are actively enrolling in the United States. We look forward to going to additional countries in the near future with this study. If you'd like to learn more, please find out at www.arvcstudies.com. Or you can go to clinicaltrials.gov and look up NCT 06228924. We're happy to say that the first patient was enrolled in the RIDGE-1 study in November. We have not released any preliminary data from this trial, but we look forward to doing so next year. This slide just shows where you can find us on social media. And please reach out with any questions. Again, patient.advocacy@tenayathera.com would be the best place to email. And I'll leave up the slide on the social media. So I'm happy to open up to questions.
Wonderful, Whit. Thank you so much. This is so exciting and encouraging, such encouraging news. We're thrilled. We have a few questions in the queue already here. Um, can you discuss natural immunity to the AAV vector and what percentage of patients have to be excluded because of that?
Yeah, so I did mention that the AAV, including AAV9 viruses, do spread among people naturally. They don't cause disease on their own, but our immune system does learn to recognize them and can form antibodies to block them. So we do measure as part of those natural history studies I mentioned levels in patients. Unfortunately, about 20% of patients have levels that would be too high to qualify for the trial. On the bright side, 80% of folks will qualify for the trial. As part of the development, we will compare patients with different titers, is the word for the level of the antibody, and learn more about whether patients with intermediate levels will also respond. So intermediate levels currently don't qualify for the trial, but could qualify potentially for future trials.
Okay, excellent. Good to know. Does the screening test for low levels, this is, I'm not sure what the sentence exactly, does the screening test for low levels of AAV neutralize antibodies?
Yes, so because we are not including patients with high titers, high levels of these antibodies, we do screen for that level. So in the process of determining whether a patient is eligible to participate in the trial, they'll work with the study doctor, get a variety of lab tests, and those will include a blood sample to measure the level of those antibodies.
Okay, all right, excellent. Has the timeline for efficacy of TN-201 been extended as a result of these recent studies?
So, we are pleased where we are, and we are progressing forward well. We are learning the time course, and there was a question earlier about how long it takes to express the messenger RNA. It does take longer than it does in mice, but that's not a surprise. That's as expected. In terms of the time course for development, our plans are to get data from the higher dose cohort. We have the opportunity to expand with more patients, one or both cohorts, to get more data. We could go up to 24 total patients for this trial. And then the next trial will likely be a phase 2/3 pivotal trial, meaning that will be discussed with the FDA and other regulatory authorities around the world as we design it to ensure that with a positive outcome, it would support approval. So we do believe the next trial we run could support approval of the gene therapy.
Wow.
We're still, just a few years off.
Okay. Yeah, it's coming faster, I think, than we thought. Back 4 years ago, we held our very first webinar with you and Tenaya, and it's 4 years ago this month, I think. So it's amazing how far things have come in that time. And someone asked, when will the next trial results for TN-201 for both cohort 1 and cohort 2 be released?
Yes, we expect to be able to release more data in 2025. It will depend on the speed of enrollment, so we can't say exactly when in 2025.
Excellent. Let's see, for PKP2 replacement, how much of an increase in PKP2 protein is needed to restore heart function? Is this strategy helpful for other gene mutations such as DSP?
I'll do them in the other order. So unfortunately, each of these gene therapies is specific to the gene that it's replacing or repairing. So for PKP2, that when that gene is dysfunctional, we replace it with a PKP2 gene. The most targeted, precise gene therapy for DSP mutations would be directed at that gene. So that's a different sequence.
Okay.
That gene is a bit longer. It's harder to fit into an AAV, so it does require more work before we can bring something like that to clinic. We certainly are working on it. We know there's a very high unmet need for DSP patients as well. And then there are other forms of genetic cardiomyopathy where it's not about replacing, but repairing or blocking. So sometimes the mutation, it's not that you don't have enough chairs, but that you have, rickety chairs that that collapse, or that, and you need to fix them. And so that gene editing is one way to fix them. And there's promising, early approaches, including some in clinic, to fix genes in human patients. And general progress in the field moving that towards the heart. And then there are other technologies to block a gene that you just need to turn off because it's causing more harm than good. So different genes each need a different technology, and some will take longer than others.
That's really a great analogy because with the chairs, because like you said, then it's not enough chairs, it's maybe a rickety chair and so forth. So I think that's very helpful for patients to understand a little bit more what's going on. And here's a question about how are the TN201 proteins made and what are they made from? Does the protein, once in the heart, regulate contraction? So like exactly, do we know what's exactly going on?
Yeah, so it definitely directly regulates contraction. So the proteins in the heart that cause contraction, you can think of a hands pulling the rope. And what the MyBPC3 protein is controlling how many hands are on the rope at any one time. And hypertrophic cardiomyopathy happens when there are too many hands on the rope. And for patients with MyBPC3 mutations, there are too many hands on the rope because the MyBPC3 protein isn't there controlling it. So we are trying to add more controls into the heart to normalize the contraction, help it relax better, in truth.
Excellent. That's also very helpful.
There was a second part to that question that I'm not sure I got to.
Oh, I think you did, actually. Yep, I think we're good. Who is the ideal patient for this therapy? Do we know yet?
So in these early trials, we are seeking patients that are highly affected by their disease today, who have the most to benefit from treatment now, and may not have as many treatment options. As we develop this, as we identify the best dose and maximize the safety of the treatment and maximize the efficacy, we anticipate that we'd be able to use it earlier and earlier in the course of the disease, with the ultimate vision of preventing disease before it starts in patients that we know have the gene mutation and have high risk of having serious disease.
Okay, excellent. So regarding RIDGE-1, in a perfect world, when do you see this approved by the FDA? I know it's hard to have a crystal ball, but
the RIDGE-1 strategy also involves 2 trials, the current RIDGE-1 trial, and then with results of that, agree with health authorities on the design of a potentially pivotal study that would support approval. So just 2 studies. Traditional drug development often has 3 phases, a phase 1 that's primarily about safety, phase 2 that starts to look at activity, and then the phase 3 is the pivotal. But gene therapy is typically faster. So again, we're still, a few years away, but, we're well on the path.
right. No, that's great. How do I know if I already have antibodies to the AAV?
Yeah, there's really no way now without participating in a clinical trial, either screening for a gene therapy trial such as ours or participating in the natural history studies like we're doing. This is not a clinically available test where you could go and ask your doctor to get checked.
Okay. Oh yes, and this is the question coming back around. What is the protein made from?
Oh, that one, right.
Yeah, right.
Yeah. So the analogy of the chairs is truthful in that it's actually the instructions that's all we're delivering is the instructions, and the parts that you need are already there. For proteins, the parts are called amino acids. You might have heard that term, but when you eat a high protein diet, it's so that your body can get all those amino acids. Your body digests the proteins you eat, breaks it into these amino acids, which are these little pieces, and then assembles proteins from those pieces, puts the amino acids together in the right order to make the protein. So the amino acids are the parts.
Excellent. Thank you. So what are potential costs associated with these gene therapies, and do you see insurance covering them at some point? What are your thoughts on the future of that?
Yeah, cost is a important topic for gene therapies. So there's a sticker shock with some of the ones that have launched in the past. At Tenaya, we are very focused on treating genetic heart diseases, many of which are more common than some of the ultra rare conditions targeted with gene therapy. So we know we need to bring the cost down. We need to bring down manufacturing costs in particular. That's an investment that we started years ago and is bearing fruit. So just more and more efficient ways to manufacture can help bring costs down. We've learned this in other novel therapies, like monoclonal antibodies now are a common type of treatment for many diseases. Used to be very expensive to make. Now, they're being used to treat cholesterol at prices that is appropriate for just lowering cholesterol because manufacturing science and efficiency improves substantially. So we're still at the very early stages on that in the gene therapy field in general, but I do expect progress. And then the other side is working with insurance companies, which do cover the approved therapies. One thing to keep in mind is that it's a single dose that will last many years. We deliver this gene to heart cells. It doesn't get diluted. Heart cells don't divide. After just 1 week of life, all of our heart cells stopped dividing. So the nuclei are stable, and we anticipate that DNA will be stable there. I mentioned the Danon study before. They've done heart biopsies 3 years after the single dose and see that it's stable. So that's consistent with animal studies and with the molecular biology. So, and it could go many more years, we will learn. So that from a payment point of view, that's also a different model. So,
Right.
industry and insurance companies will partner with the best ways to make this accessible for as many patients as possible.
Right. Absolutely. No, it's very interesting. And how does the dosing work? So I know it's one dose, and people are asking in various ways if one dose it sort of wears off after a while. I don't even know if that's possible. It can wear off. Would people be eligible for another dose? Obviously, we've talked about probably not. How does that work?
So we don't expect it to wear off for years, but we don't know for sure. And that is something we will certainly track. But again, it doesn't get diluted out. It's in that nucleus of that cell, and that cell isn't dividing, the nucleus isn't dividing.
Right.
The beta V stable viruses have evolved to be able to do that. And we're taking advantage of some of the structure in the DNA that the viruses use. So we don't expect that to wash out at all. But redosing for AAV-based gene therapies is not possible with today's technology. There's a lot of work on that. There are other diseases, specifically are often diseases for proteins made in the liver, like hemophilia and so forth. The liver cells do divide, and it does dilute out the effect of gene therapy over time. So there's a lot of interest in the field about how to a dose in a way that will be effective in patients that have already developed neutralizing antibodies and ways to prevent those neutralizing antibodies. A lot of science going into it. Not, we don't know if and when that will work. I'm cautiously optimistic, but I don't think it's going to be, next year or the year after.
Right, not right away. And I want to be cautious about some people think gene therapy is a cure. How do you address that language? It's not technically a cure, is it?
And people kind of just, the definition of cure is variable. When we talk to experts who think about, what defines a cure, I guess I would say it's close. It's kind of got a lot of the features that you'd want. Like it was a one-time dose and you don't have to think about it. And it directly fixes the underlying cause of the disease. by replacing the gene that's not working. So it's on that spectrum of different types of treatments. We're not just reducing a symptom, and it's not an indirect thing, and it's not something you have to keep taking. It's not like something that that the disease comes right back if you stop, that kind of thing. So it is closer on the spectrum towards cure.
Okay, excellent. And you talked about that it will be available in other countries. Do you have any time frame for that?
So for MYBPC3 HCM and the MyPeak-1 study, that is a US-based study, and we look
Yep.
forward to going to other countries with the next study, the pivotal study that I talked about. And that we still need some months to a year to get towards that direction. On for the Ridge-1, that study will become an international study, and we look forward to announcing that sooner rather than later.
And that's exciting, so exciting. So when would you consider someone ineligible for treatment? Do we know yet, is there an instance where their heart failure is too advanced and they would not be a candidate?
Yes, we do. I think in theory, there probably is a point at which heart failure is too advanced to respond to these types of gene therapies, and specifically when there's too much fibrosis. So cardiomyopathy leads to a process called replacement fibrosis, where the heart muscle cells or myocytes are lost and replaced by scar tissue and fibroblasts. And those fibroblasts can't respond to gene therapy. So we don't think we can reverse fibrosis. And at some point, unfortunately, there are just too few myocytes remaining to respond to a gene therapy. We don't know what that point is. We are, I'd say, conservative about that in the initial trials. So for patients that have significant loss of heart muscle cells, they're not eligible for the early trials. But later in development, we will test it in a broader range of patients to see whether patients further along in their course will also respond.
No, that would be wonderful. We had a couple of questions came in that are very specific about the PKP2 trial and what's required with the trial. How far do you have to be from a trial site? What's the procedure? What's the process? I would like to refer them back to reach out to Wendy with their questions. And I will also follow up with this webinar recording in an email tomorrow. And I will also include information how to contact Wendy about these trials. So I do recommend people follow that route unless you had any other feedback on that right now.
Well, thank you to the folks asking the question. Thank you for considering participating in research, and thanks to all of you considering participating in drug development in any way that can. I'm glad to hear your interest in our clinical trials. That there is a process to get in, a process where we can introduce you to a study doctor who can first go through your specific medical history and condition and determine whether you meet all of the eligibility criteria. Some of those are listed on the websites and clinicaltrials.gov and our website, so you can get a preview. But the list is longer, and so that investigator can go through with you. Another key part of the discussion with the investigator is discussing what is known in far more detail than I can here about the gene therapy, what the risks are, what the possible benefits are, the fact that we don't know if it works yet or not, and go through all those pros and cons. And of course, the schedule and the assessments and the timing of those to see if that would fit with your schedule. So, it's a good, robust discussion, usually over more than one visit with a study doctor.
Excellent.
We do support travel. And in fact, we've had patients already traveling substantial distances, different states, and so forth, to participate, and Tenaya reimburses those expenses. But there are a large number of study visits. So like many patients may know of late stage trials where the visits are infrequent, but for an early study with such a focus on safety, there is a fair number of visits, particularly in that first year.
Excellent.
Just to say, the trial is not for everybody, but it's critical that people participate, and we are extremely thankful to our patient participants, clearly heroes that are advancing this novel therapy for everybody, for all patients.
Excellent. One person just put in the chat, excellent presentation. I am interested in participating and learning more. And I would just reiterate your prior slide. And I will also, actually, if we go to one next slide, Justine, I think there it is at the bottom. Anyone interested, please reach out to Wendy and her team. You can contact them at patient.advocacy@tenayathera.com. You see it at the bottom of your screen there. I will also send that in the follow-up email tomorrow. Whit, we just can't thank you and Tenaya enough. Very exciting news, and we're so thrilled that we're 4 years into the future from where we started with you. So we can see this all coming to fruition now. So thank you for your partnership and your support. And yeah, we look forward to what next year brings.
Absolutely. A lot has changed on our end. A lot has changed on your end as well. So thank you for everything you're doing.
It has. Thank you so much. And thank you to Wendy and Lori and your whole team. Excellent. Justine, I think we'll go back to our deck and I will wrap up. We have some thank yous and some exciting information coming. So first, thank you to all the DCM Foundation and GCAC sponsors whose logos you see there at the top. Also, thank you to all of our patient partner groups. We continue to expand and have new groups joining our efforts to share the message about the importance of genetic testing for cardiomyopathy. And we're thrilled that all of those companies help support us all year long. We work hand in hand together with them to get the word out. Next slide. So again, if you're interested in learning more about Tenaya Therapeutics, you can go to their website, tenayatherapeutics.com. You can also go to hcmstudies.com. I will include that information in the follow-up email. For the DCM Foundation, you can check that out at dcmfoundation.org. And anyone interested in genetics and cardiomyopathy, please go to geneticcardiomyopathy.org if you haven't been there before. Great website. There's a ton of information. If you have questions, you can reach out to me. My email address is jrock@dcmfoundation.org. Next slide. We're thrilled to announce our next webinar will take place on January 22nd, 2025 at 6 p.m. Eastern Time. We are going to announce the launch of the Genetic Cardiomyopathy Registry. This is a huge accomplishment. It took us 12 months with a lot of hard work in partnership with NORD, and we will tell you all a lot more about it. Come join us on January 22nd. The registration information is up on the DCM Foundation website, and we look forward to seeing you there. Or if you can't attend live, just register and you will get a copy of the recording. Next slide. Thank you again. We appreciate Tenaya tremendously. Can't thank you enough for your partnership. Justine is working behind the scenes with the technology as always. Thank you so much, Justine, for your help. And happy holidays, everyone. Stay safe, be well, and we will see you in the new year. Thanks, guys. Take care now. Bye-bye.